So it's my great pleasure to introduce today's speaker, Dr. Samer Hattar. Dr. Hattar is the Chief and Senior Investigator of the Section on Light and Circadian Rhythms at the National Institute of Mental Health. And prior to joining the NIH, he was a tenured associate professor at Johns Hopkins University in the Department of Biology and Neuroscience. Dr. Hattar received his PhD in biochemistry from the University of Houston, followed by postdoctoral training at Johns Hopkins School of Medicine in the labs of Dr. King-Wai Yao. It was during this time that he researched the role of melanopsin-containing photoreceptors in non-image forming functions. His research has been supported by innumerable grants through the National Eye Institute, National Institute of Mental Health, and the NIH. And his work has led to over 80 publications and book chapters in esteemed journals such as Nature, Neuron, and Science. In addition to his scientific contributions, Dr. Hattar is frequently a keynote speaker worldwide at esteemed institutions, has chaired and organized major international conferences, serves on editorial boards, including Science Advances and PLOS Biology, and is a leader in retina research. Today, he will be speaking to us about the retina's interaction with the circadian pacemaker beyond photoentrainment. Please join me in welcoming Dr. Samer Hattar.
Okay, thank you very much for the invitation. I'm really excited to be here. Thank you, Regina, for the invitation and for the correspondence. Today, I'm going to go beyond the role of the retina for vision and actually beyond its role for even in training the circadian oscillator. And hopefully, I will show you at the end with the punchline that actually circadian time is not only measured in the brain, but actually start being measured in the retina. And that's what's very exciting about today's talk. But before I would start, I would like to thank the people who helped me do the work. This work started at Johns Hopkins University, where we were part of the mouse dry lab, where myself, Hai-Chen Zhao, and Reggie Cruvilla, who happens to be my wife, we were part of the mouse dry lab, where we're 3 mouse neuroscientists, Sherry agents, students, and postdocs. And this is great for young scientists because it allowed us to go beyond our little area of expertise. And then I just moved to the NIMH in 2017, the National Institute of Mental Health. And there was a big change going from an undergraduate to the National Institutes of Health because my lab changed from graduate and undergraduate lab to more postdoc lab. But I have been enjoying the institution there. I shouldn't be saying this, but till recently. And you guys know why, I think. So the rotation of the Earth around its axis produces a very predictable change in the light-dark environment. That is really good if you want to know the presence or absence of light. So it's really good for you to know if it's day or night. But this really produces a really big conundrum. So for example, if you take 1 centimeter square of your skin and you look in a very sunny day, like California, and in a centimeter square per second, you get 10 to the 16 photons. That's not a mistake. That's actually a true number. The same centimeter square at night, you get a few hundred photons. So that causes a problem for vision. How can you see across this amazing intensity? And I don't have to tell you because you guys are the expert. This really is achieved by the amazing adaptability of the rods and cones photoreceptors across that intensity. So this is really interesting because you have a problem for vision, which is great, but there's also a predictability for the light-dark cycle. But vision is not the only thing that light allows you to do. So for example, in mammals, all light detection was centralized to the eye, but in non-mammalian vertebrates, actually there are organismal effects of light that are not related to the eyes. For example, in these birds, detecting of the day length is dependent on non-ocular photoreceptors. And just to show you the importance of light beyond image formation, here is a male bird, just put on short day versus long day. So only difference is the length of light in the day. And look at the size of the gonad of the male. This is what happens when they are under long day versus short day. So the mating and the reproduction of this bird is dependent on the day length. And why is that important? Because you want them to get their progeny at the most optimal time in the year so they could survive. So light is not only important for vision, but it's really important for the reproduction and the survival of organisms. This is an ophthalmology department, so I don't have to convince you that as primates, we really love vision because this is the thing that we appreciate most. And usually vision starts in the retina, and the retina analyze the information and send in parallel information to the thalamus, and then the cortex reassemble that information, and we consciously see this image. So even though this is a two-dimensional image, you could see the waterfalls, the cliffs, and everything. And this happens, and I don't have to tell you that, because the retina is really beautiful. It's an inverted structure, so light passes all these neurons before it hits the photosensitive parts of rods and cones, which contain rhodopsin and conopsin. Then through horizontal and vertical signaling, this information is sent through different streams to the retinal ganglion cells, which then send this information to the brain. However, the retina also detects light for functions that are not associated with image-forming functions, which include circadian photoentrainment, which is actually mostly controlled by the circadian pacemaker located in the suprachiasmatic nucleus. And I don't have time to show you the data, but also we think the intergeniculate leaflet is a very important part of this system. I'll be happy to answer a question later if you have any. And the pupil light reflex. And I would like to use the pupil light reflex to show you that how seemingly simple behavior can be so complicated. So this pupil light reflex could function just as well by having a photopigment in the iris muscle and be controlled locally. But in fact, this system is so complicated. So the light has to hit the retina. It has to go to a region in the pretectum known as the olivary pretectal nucleus. Then it has to go to the Edgar Westphal nucleus. Then it has to go to the ciliary ganglia, the ciliary muscle, then drives the pupil constriction. And the theme of my talk today is going to show you that although non-image forming functions look seemingly simple, you'll find that they are just as complicated as image forming functions, and that light has parallel pathways that affect different behaviors through different IPRGC population. So now we know, based on many different behaviors, that the eye detects light for vision, but it also to entrain the circadian oscillator located in the SCN. And in the past, and actually even till recently, still people think that all light information to regulate our clocks located across the whole body has to be mediated through the SCN. And I'll get back to this information later. So how do we model circadian photoentrainment in the lab? It's actually very simple. We have these, what we call actogram. Really, they are activity graphs. So what we do, we put a mouse in a cage with a wheel. Every time the mice run on the wheel, we record the activity. So for example, here is a very short activity. Here is a very long activity. And you put the mice under multiple days, and you change the light-dark environment. So for example, the first 10 days, the mice are under 12 hours of light, 12 hours in dark, and you double plot the data so you could see the onset and offset in the same light of 2 successive days. So what you notice is that mice under light-dark conditions, if they can photoentrain to the light-dark cycle, they have an exact 24-hour period. You could see the rhythm is beautifully 24-hour. And their phase relation is associated with the darkness because mice are nocturnal animals, so they start running when the lights are off. However, when you put them in constant dark conditions, what you notice that the clock is still running, it's beautiful, but it's not exactly 24 hours, hence the name circadian. Circa means approximate, and dian is day. So what light does, it makes this circadian clock exactly 24 hours. And you think that's simple, but that's a very complicated process, as I'm going to show you in a second. Interestingly, in a human, the circadian clock is longer than in 24 hours. So if you have a normal circadian clock and I put you in constant conditions, your sleep-wake cycle will be going in this direction instead of this direction, okay? By the way, feel free to interrupt me. I'll be happy to be interrupted if you have any questions, because if you lose any information, you're not going to enjoy the remaining of the talk. So if you have any simple questions, I'll be happy to answer. So because of the circadian field and the availability of amazing mutation from the retinal field, it was really these beautiful 2 science papers from Russell Foster group that really opened this field wide open. So before 99, if you told anybody in the retinal field, circadian field, that there are photoreceptors distinct from rods and cones in the mammalian retina, they would have laughed at you. But in 1999, there were two papers published in Science. And by the way, the papers that were published from other labs will be on the left, and the paper that are published from my lab, they'll be on the right, where this group produced animals based on the mutations from the retinal field that completely lack rods and cones, and hence they were called rodless, coneless. As you would imagine, these animals were completely image blind. However, when they were put under circadian photoentrainment, they looked no different whatsoever from the wild-type animals. However, when these researchers took the eyes out of these animals, then they free ran as if they are in constant conditions. So then they suggested that there has to be some photoreceptors distinct from the rods and cones that are found in the inner retina of the mammalian, retina. And to make a long story short, it was really the pioneering work of Ignacio Provencio, and then in collaboration with us, that we now know that there is a subset of ganglion cells that are intrinsically photosensitive. That's a fancy way of saying they are photoreceptors because they express a photopigment melanopsin, which is similar to rhodopsin and conopsin. And so I'm going to refer to these ganglion as intrinsically photosensitive retinal ganglion cells, or IPRGCs, okay? All right, so now we know that the mammalian retina, in addition to the conventional ganglion cells, have these third type of photoreceptors. These photoreceptors are not specialized like rods and cones. They are actually neurons. They don't have a modified cilia or a modified rhabdomere, and they respond to light intrinsically, but they could also receive rod and cone input from the outer retina, just like normal ganglion cells. So they have a dual personality. So what happens then if you knock out rod, cone, and melanopsin function? Is there more surprises? No, luckily there aren't any, because when you knock out the three photoreceptive system, you get a free-running rhythms. And I'm sure you're hearing about neuroopsin 3 and neuroopsin 5. Neuroopsin 3 and neuroopsin 5 can entrain local clocks, but they cannot entrain organismal circadian photoentrainment. For organismal circadian photoentrainment, it's all the retina. So now we know then that even in the absence of melanopsin, rods and cones can signal through these ganglion cells. So it wasn't surprising at the time that when we did melanopsin knockout animals, just like the rodless, coneless animals, they also entrained similar to the wild-type animals. And in fact, if you look very carefully, when rods and cones are functional, melanopsin IPRGCs have much better response to light, and they are more faithful to the response of light. And that really bothered me as a scientist. I'm like, wait a minute. If rods and cones make this cell respond better to light, why is melanopsin conserved across evolution? Why do we look at every vertebrate and melanopsin has not been eliminated? And the example I thought about is the olfactory system. If you look at our olfactory receptor genome, it's a graveyard of destruction of mutations. Our olfactory system is disastrous compared to the mouse or the dog or the cat. So I wondered, is there a function for melanopsin, phototransduction pathway, that rods and cones cannot compensate for? And really, this work was done by Cara Altemus in the lab, and we first found it in sleep. So instead of now looking at the activity, now I'm showing you the sleep EEG rhythms. And so what you're looking here, you're looking at REM sleep, and the spikes are, the flat is non-REM sleep, and the spikes are REM sleep. And you could see that the mice sleep mostly in the day, but they also take nap in the night when they are active. And so just as you would imagine, they beautifully entrain to the light-dark cycle, and they sleep mostly in the day. Different than us, they don't sleep continuously. They have interrupted sleep. So what Cara did a very simple experiment. She entrained them to 12-12 light-dark cycle, and then 2 hours after darkness, she came and gave a very bright light, 3 hours in the night. And it was really remarkable. The minute she gave the 3-hour light pulse, sleep was increased at night. So there was a direct effect of light on sleep. And it's not surprising, like if I dim the light really dim here in this room, even you may be enjoying this talk and I have a black background in my slides, you will start feeling sleepy even though it's the day. So there is clear effect on the environment on your sleep directly. But what was really exciting for us, if you do the melanopsin knockout animals, the sleep was entrained to the light-dark cycle, but now and even though rods and cones can signal through IPRGCs, but now the direct effect of light was completely abolished. So that tells you that even though rods and cones can signal through these cells, they cannot compensate for the absence of melanopsin. The problem, this is sleep, so we wanted to see if it's given to other functions as well. It's not only for sleep. So Alan Ropp wanted to do it in body temperature, which is also have a beautiful rhythms in mammals. So here you could see body temperature. Again, it's low in the day, high at night. And when Alan came in and gave the same 3-hour light pulse, you could see that body temperature is acutely reduced for the whole duration of the light pulse. So people forget the power of the retina. All you have to do is shine light on the retina and your body temperature in the mouse is decreased for the whole duration of the light pulse. It's just remarkable. This is the wild type. This is the melanopsin knockout animal, beautifully entrained to the light-dark cycle, no problem whatsoever, no reduction in body temperature whatsoever. Rods and cones fail miserably at compensating for the melanopsin phototransduction pathway. Again, this was very exciting to us, but when do you get 3 hours of light in the middle of night in the environment? Luckily, I had a very smart student, Melissa Simmons, who said you get that when day length changes. That's the time that you get light more in the night. And the problem, as circadian biologists, we took the easy way out. We gave animal 12 hours of light, 12 hours of dark. That's a perfect light-dark cycle. She said, what would happen if we give them 16-8 light-dark cycle? Just change the amount of light that you give them in the day, just like the bird experiment that I showed you before. Remarkably now, the melanopsin knockout animal all of a sudden has major problem in entrainment. So we sent this paper to Nature Communication. The reviewers loved it. One reviewer said, I would love this paper, but you need to explain to me, why do rods and cones fail when you add 4 hours of light? I said, if I know this, you think I would have sent the paper to Nature Communication? Honestly. Anyways, so we still haven't published this work because we're going to figure out why rods fail with more 4 hours of light. I will never figure this out in my whole life. I need the new generation to figure this out. Anyway, so because these are ganglion cells, we also labeled them just like the olfactory system to trace them to the brain. And what was really remarkable is that, as you could see here, they project to the circadian oscillator very beautifully, but they also project to other areas in the brain that regulate other function distinct from the circadian system, including pupil constriction, mood regulation, and sleep and alertness. And furthermore, work from these 3 amazing women in the lab have shown that IPRGCs are very diverse. In fact, now there is an M6 type as well, which I didn't update here. And they are at least differentiated based on the expression of melanopsin, with the highest expressor being the M1. That's why this is the one we first identified and we worked most on. But they are different in their arborization in the IPL, different in their rod cone input, different in their sensitivity to the light, and so on and so forth. And in fact, even before we discovered the diversity of IPRGCs, Ali Gouler and Jenny, you've seen in the previous slide, decided to ask a very simple question. What would happen if we kill IPRGCs? Can the animal entrain to the light-dark cycle in the absence of IPRGCs? And so to do that, they expressed the attenuated diphtheria toxin under the melanopsin promoter. And it was very lucky for us that we expressed the attenuated form of diphtheria toxin, because this diphtheria toxin, before we even knew about these population, we discovered later that it only killed the M1 population and only killed it after 2 months of the animal's birth. So we literally, by luck, produced an animal where we killed only M1 cells and only killed them conditionally after development. So when we looked at these animals for vision, development, and function, they were completely unaffected. So now we produced an animal different than the animal I showed you before, an animal that is image normal, but has no IPRGCs and only one type of IPRGCs, the M1. And I'm honestly still stunned with the data of this animal because something as important would depend on one single population of iPRGCs. These animals completely failed, even in 12-12 light-dark cycle, completely failed to photoentrain to the light-dark cycle. So you could see, again, the wild-type animals follow the light-dark cycle very beautifully. These animals free run similar to the triple knockout animal. So this was great because now we could study vision and circadian photoentrainment in different pathways, but it also tells you that evolutionary circadian photoentrainment and vision must have been separated at the level of the intrinsically photosensitive iPRGCs. Yes, Chris?
And this is also without rods and cones.
No, this is rods and cones functional, and the animal can see. You do all the image-forming functions, the animal completely normal. So it's like, when I used to teach my undergraduate at Hopkins, I used to say this is like having a mathematical woman, asking her, what is the most complicated equation? And she solves it to you in 5 minutes. And then you ask her, what is 1 + 1 equals to? And she's like, I don't know. So like you could solve a very complicated image-forming functions, but you can't tell when is the day and when is the night. It's just really mind-boggling for me. And even more interesting in a way that like the nature gave us a gift in a way, we found that only the SCN projecting iPRGCs do not express the transcription factor BRAIN3B. All the remaining M1 iPRGCs and all non-M1 iPRGC express this BRAIN3B transcription factor. So Alan Chen, when he was in the lab, decided to do intersectional genetics and use melanopsin Cree line in combination with BRAIN3B stop DTA line to kill all iPRGCs that express BRAIN3B. And when he did that, we were able to kill all non-SCN projecting iPRGCs without touching the SCN projecting iPRGCs. And just to show you how beautiful this animal, you could see the SCN is still fully innervated. The image-forming centers are still fully innervated, but look at all the non-SCN projecting iPRGCs, the areas are completely denervated. And because the SCN is completely innervated, circadian photoentrainment was completely intact in this animal. So now we also have an animal that have normal image functions, that have normal circadian photoentrainment, but lack the innervation to other non-SCN brain regions. So then we wanted to ask a question, is this thing correct? So now we know that M1 project to the SCN and control circadian photoentrainment, but is it possible that the eye can also control these functions independent of the SCN? So to do that, we started looking again first at body temperature. So here's heterozygous or wild type. Again, I showed you before that these animals photoentrain and they respond beautifully to a light at night for body temperature. Now we're going to use the animals where the SCN is fully innervated, but the other ganglion cell, the other iPRGCs are ablated. And it's just remarkable. Again, you could see the animals are photoentrained, but again, you don't get the induction by light. So it means that the SCN is not required for the direct light effect on sleep or body temperature. They are different pathways. So then this made us wonder, is this direct pathway controls more than sleep? And if we didn't have data on all of this, I wouldn't have put it there. So we asked a very simple question. Can we figure out if the direct pathway can regulate all these functions there? And unfortunately, all of this, fortunately, is published, but unfortunately, I don't have time to go through it, but I'll be happy to discuss it. So I'll just go quickly to show you what we found. We found that through the SCN in a clock-independent manner, light can affect the hippocampus to affect cognitive functions. Through the intergeniculate leaflet in the visual thalamus, through the SCN in a clock-independent fashion again, it can affect time-restricted feeding and energy and metabolism. I'm going to show you later through the preoptic area, it can affect only non-REM sleep, but not REM or body temperature. And then through an area that we discovered specifically for mood regulation and the prefrontal cortex, it can affect mood control. And recently, we also even shown that it can affect social behavior. So we think that light is so interesting, even in a nocturnal animal, that it has this circadian photoentrainment pathway, which I'm going to talk about in the remaining part of my talk, but it also has this direct pathway that affects all these different behaviors. So because of that, we decided to change our thinking in the field, even as a circadian biologist. This is really an important path, and I'm going to be talking about it the remaining part of my talk. But one has to think about the possibility that light can directly affect the homeostat, all the behavior itself, directly independent of the circadian pacemaker. And in fact, when you look at the literature very carefully, you find that there's already papers published in 2009 where you know that light can affect the homeostatic part of the sleep directly independent of the circadian pacemaker. So let me show you why this tripartite model that I showed you here is important. So people know that sleep is controlled by two processes, a circadian process and a homeostatic process. Simply put, the circadian process tells you when you have to sleep, and the homeostatic process tells you how much you have to sleep. So the more you stay awake, the more you have to sleep. And before we thought that light can only go through the circadian process. But recently, a work from Zizang and Karim Bayer in the lab, we found that the preoptic area that regulates sleep has three different populations of neurons that inhibit wake alerting center of the brain. But only one of these populations, the corticosteroid releasing hormone population, received direct light input from IPRGCs for light to regulate specifically non-REM sleep. Let me show you how we found this. We actually used the melanopsin Cree line, and we injected retrograde virus that will only express the dread, activating dread, in the presence of FLIP and Cree. And then we injected an AAV2 virus that will only express FLIP in the presence of Cree. So only cells that project to the preoptic area will be able to express dread. And now you measure the non-REM and REM sleep in the animals, and what you could see here when you add CNO, you get a beautiful increase in non-REM sleep, but not REM sleep. So that tells us that there is a different circuits even for regulating different aspects of sleep by light. So that tells you how important the light environment is for regulating behaviors. So in the remaining part of my talk, I'm going to be telling you about how the eye is actually part of the circadian pacemaker. And this really, we started getting that information in 2017, a paper that was published in eLife, beautiful paper by Kylie Chu. So remember I told you when we did the ADTA animals, we only killed the IPRGCs in adult animals, and that's why the period length was not affected. However, we noticed if we use a strong form of DTA, which killed the IPRGC early in development, now the circadian period is modified. It actually becomes like a human. It becomes longer. So this gave us an idea that maybe the retina has a bigger effect on the pacemaker and that we didn't appreciate before. But before I show you the data, I have to tell you one thing that is really interesting about the circadian clock, which is known as the phase response curve. So if you put animals in constant darkness, you don't give them any light dark cycle, and you give them a light pulse early in their evening, you get a phase delay. Why? Because the animal think there is still sunlight, I'm thinking it's early night, I should delay my rhythm, so they delay it and it persists. If you give them the same light pulse in the day, you don't get any phase shifts. Now what's amazing, if you give the same light pulse later in the night, they think the day is coming earlier, and now you get a phase advance. The same light pulse produces phase advance. Now, if you draw the whole response across the whole day-night cycle, you get what is known as a phase response curve, or PRC, where you get no phase responses in the day, phase delay early at night, then it switches to phase advances late at night. And people, including myself in the circadian yield for 50 or 60 years, thought that all the ability of this phase response curve to be observed has to be determined by the brain, by the SCN. It has nothing to do with the retina. And honestly, if you think why, it's because of all this data, but I want you to concentrate on one paper I'm going to show you that really convinced me 100% that this is the case. This is a paper in Science in 1994. And this paper, the author isolated the SCN in a dish, and they added glutamate, which comes from the ganglion cells of the retina. So nicely, the SCN in a dish have a beautiful rhythm in electrical activity. And the electrical activity is high during the day and low during the night. So what they did, they gave glutamate, tons of glutamate at early night, and they got a beautiful phase delay in the electrical activity, just like the animal. They give glutamate late in the night, they got a beautiful advance. They get glutamate in the day, they got nothing. And when they draw the phase response curve to glutamate versus light, they were superimposable. There was no way that completely convinced the whole field that the gate has to be in the SCN. Even our work in 2020, here is a rhythm in constant darkness. You could see that the phosphorhistone 3 is completely not expressed in the SCN, phosphorylated form. If you shine light in the day, this histone 3 is not phosphorylated on serine. If you add it in the night, you get a beautiful phosphorylation of serine consistent with the phase response curve. However, Corinne and Rucci decided to tell me in one day that they think there is a gate in the retina. And I said, I'm a circadian biologist. You're wasting my time. I swear to God, I'm not lying. I bet for them 10 to 1 that they're just going to waste my money and they will never discover a gate in the SCN. But I didn't know they were smarter than me and they read the literature very well. The reason they thought that, because Michael Doe's group published a very beautiful paper in Science and Cell, where they found that iPRGCs at the single cell level, they have what is known as depolarization block. So if you look at an iPRGC, they respond beautifully linearly to first increases in light intensity, but then they actually stop responding a little bit and then they shut off completely. So this is what they call an inverted V response, because they respond linearly first, then they shut off. So as a population, iPRGC respond to a very wide range of intensities, but as single cells, at every intensity, only few iPRGCs respond to light. So Corinne and Rucci thought that maybe this is a gate, that maybe this is evolutionary, was intentionally designed to prevent the SCN from getting too much light information. And they thought if we can activate iPRGCs all at the same time, we may be able to break the SCN gate. And to do this, they used the melanopsin Cree, and they simply injected a virus that will express activating dread under the control of Cree. But before we did that, they wanted to make sure that CNO works, so we collaborated with Jeff Diamond Group and his postdoc Amerta Na. And you could see very nicely that every iPRGC we recorded from, when you add CNO, you get a beautiful firing in these iPRGCs. So the system works. So now let's see what happens when you do that in the day. So as I showed you many times, if you give light in the day, when the SCN gate is closed, you don't get any phase shift. And honestly, I was fell off my chair when they showed me the data. If you now do CNO, not only you get a phase shift, you get a huge phase shift, a 6-hour phase shift. Just to put it in context, even when the SCN gate is open and you give light at night, you only get 2-hour phase shift. And if you do CNO at night, you don't get any more. Once you break the gate, you break the gate. You get also 6-hour phase shift. So then I said, why didn't the people who did the glutamate in the SCN work? Why didn't the experiment work in the SCN in isolation? It should have worked. Part of the reason it didn't work is because the LGN, the visual thalamus, is important for this function. It's not important for the phase shift at night, but it's important for the phase shift in the day. And this is how we did this. We actually used the brain 3B animal, where you have the SCN innervation without the innervation of the other brain regions. And if you look at the nighttime activation with CNO, you get a beautiful 6-hour phase shift in both the iPRGCs when they are all present, or where only the SCN ones are present. But look what happens during the day. You only get the big phase shift when all iPRGCs are present, but you only get the ones that are only 2 hours, similar to the light, when only the SCN is present. And I'm not going to show you the data. We also found that in addition to the different circuit, the neurotransmitter PACAP is also required, so glutamate is not sufficient. So now we know that in addition to the SCN gate, remarkably, there is a retinal gate that is caused by the depolarization block in IPRGCs that help to prevent light from destroying the SCN gate. So we sent this paper to Cell, and one of the Cell editors said, this is great, but it's all CNO, it's non-physiological, which is true. So I was complaining to one of my colleagues that, yeah, we didn't have a way to do it with physiology. It'd be nice if we could activate melanopsin enough, but not cause depolarization block, and that was my only idea in the paper. So I thought maybe we could use the wavelength of light to somehow activate melanopsin sufficiently enough, not cause them to have depolarization block. And clearly we used the melanopsin absorption maxima. We thought the blue would be great for activating them very strongly and cause depolarization block. We can't use longer because then they are not very efficient. But what about if we use the UV light? UV is not only weak at activating melanopsin, but maybe the OS cone can also contribute to the function. So consistent with what we know, if you use a blue light, you could see you don't get any phase shift. But we were stunned that if we use a UV light, you get a phase shift. Yes, you don't get a 6-hour phase shift. It's not as efficient as the CNO, but you still get a breaking in the gate in the daytime. And then we collaborated again with Jeff Diamond, and we recorded from the same IPRGCs under the 2 different colors, and it was remarkable what we found. Blue light always caused depolarization block earlier, and it's a narrower range. So if you look at the range of responses under blue light is much narrower than under UV light. Then we wondered, the sun has a lot of UV light. Can we use the sunlight to actually cause this to happen? So then when we literally took the animals outside the lab and put them in sunlight, and you could see that mice don't like sunlight. So after we exposed them to sunlight for 3 days, they didn't like to run on the wheel. But remarkably, the phase was still completely the same before and after the sunlight stimulation compared to the UV light. When we then collaborated with Jeff Diamond Land, we noticed that what's remarkable is that the SCN under sunlight is actually thinking the animal is in the dark because the IPRGCs are completely depolarized blocked. Literally, the sunlight is so strong, the IPRGCs are not signaling. So now we think that actually there are 2 gates in the animals. You have the circadian gate in the SCN, which is regulated in a day-night manner. But we have a homeostatic gate in the IPRGCs that is present across the day-night cycle. And remarkably, the pathway that drives the day phase shift versus the night phase shift are different. With the day phase shift requires the LGN and the neurotransmitter backup. But despite the fact that we figured out the molecular contribution for the retinal gate, we still don't have any idea what is the molecular gate in the SCN. So to do that, we actually decided to do something that surprisingly the field has not done yet. So the field has sequenced the SCN multiple times in constant darkness, or they gave a light pulse at the sensitive portion of the cycle. But nobody has given a light pulse in the insensitive part or the phase advanced part of the cycle. So this is what we did. We decided to not only sequence the SCN in the constant dark, but also give light pulse at all the phases of the phase response curve. And what we found, similar to what the field found, is that the SCN is very interesting. It has 10 different types of neurons. But what I want to concentrate on is a couple of very interesting observations. First, the dead zone is really not dead. Actually, the dead zone is the most responsive part of the phase response curve. So light really affects the SCN during the day. So why you don't get a phase shift during the day? Because the pathway that produces a phase shift requires the cyclic AMP and the Krebs phosphorylation. So this is actually a paper from David Ginty and in 1993 in Science, showing that Krebs phosphorylation, just like the phosphohistone 3, is only observed at CT19, but not at CT6. So when we looked at which genes are affected when we give light at different phases, we noticed that during the day, you get mostly synapse organization. But during the night is when you get cyclic AMP signaling. So the gate is very specifically to a very specific component of the pathway. So then said, what kind of molecules we get that are affected in the light-dark cycle? And what we found is this very interesting molecule is phosphodiesterase 10A. And why did we get excited about this? Because phosphodiesterase 10A could be a brake system on the cyclic AMP pathway. So then Rucci decided to look, how is the rhythm of this molecule? Does it correlate with the PRC? So she looked at the protein level of this molecule, and it was amazing. This PDE10 is very high in the day, very low at night, and intermediate at CT22. And what you notice is when PDE10 is very high, you don't get any response to light. When it's very low, you get a big 2-hour phase shift. And when it's an intermediate, you get a weak 45-minute phase advance. So we said, then what would happen if we knock out this gene? The prediction is that the gate in the SCN should be broken. And without breaking the retina gate, you should get phase shift in the day. So this is what we did. That's the prediction. This is what we did. So we got a PDE10 knockout. And you could see that the phase delays were not affected because it's not expressed at night. But remarkably, now with simple white light, you could actually produce a 1-hour phase shift during the day, similar to the UV light we did. So we got very excited because now we have a way to figure out why did nature produce gates in the retina and the SCN. Finally, we could figure this out. So we just put this animal under light, dark, and dark, dark conditions. And look what happens. I'll show you the data. Control animals, they have a beautiful rhythms both under light, dark, and under dark, dark conditions. While the PDE10 knockout remarkably have a beautiful rhythm under DD, but really they have miserable rhythm under light, dark conditions. And this is hard to explain for people outside the vision field because they think once you're in light, you're in light. But you guys know that anytime you move your head, you get different intensities of light. So the gate is preventing you from getting different intensities and shifting your clock every time you go from one room to another. And that's why you needed a gate to prevent this from happening, so you don't mess up your clock in the daytime. So now we came up with a new idea that actually the SCN is not the only circadian pacemaker, but we think the retina and the SCN constitute what I'm going to call an integrated pacemaker, where a homeostatic gate in the SCN and a circadian gate in the a homeostatic gate in the retina on the IPRGC level and a circadian gate in the SCN really form an integrated pacemaker to help the animal control the entrainment to the light-dark environment. I don't know if you can indulge me with 7 more minutes to just tell you one last story. Yeah. So this is the last story that this is unpublished, and there's this other story now that is unpublished. So all this work that we have done so far mostly concerned the M1 population. So we wanted to know what about the other non-M1 population, and this is work now by Kat Daly in the lab, where she took all the sequence information that is available for IPRGCs, and she found that M4 cells actually constitute the alpha on ganglion cells. And she said, can I label these population very specifically? So she looked at the sequences and she found that paravalbumin and melanopsin can be a way to label the M4 independently of the other IPRGCs. So to confirm this, she stained with melanopsin and SMI32, which labels on alpha ganglion cells and paravalbumin, and she confirmed the sequencing data that the 3 co-localize with each other. So then what she did an AAV virus that will express YFP in the presence of FLIP and CRE, and she mated an OPN CRE paravalbumin FLIP line. And we were so happy. You could see here that she specifically only labeled the M4 cells in the retina. So now she's only labeling the on-alpha M4 IPRGCs. So then she wanted to trace this population to the brain. Since they are the M4 ganglion cells, you could see they completely avoid the SCN, but they beautifully innervate the rhythm areas, the vision areas. I'm so obsessed with the rhythm. The vision areas of the brain, the dorsal lateral genicular nucleus, and in fact, they completely avoid even the rhythm area of the brain, the intergeniculate leaflet. And if you look at all their innervation pattern, This iPRGC is actually an image-forming iPRGCs. It doesn't contribute at all to non-image-forming function. So now, like, we expanded the role of iPRGCs from non-image-forming functions to specifically for image-forming functions. So then we wanted to manipulate this population and see what is its role in driving behavior. So we did two things. We expressed either an inhibitory dread in it or an excitatory dread. So you could see we could either excite them or we could inhibit them in the presence of light. So first of all, we looked at circadian photoentrainment. Nothing happened because they don't project to the SCN. But even the pupil, they don't contribute at all to the pupil constriction. But what about contrast detection? Because we know that M4 on alpha ganglion cells are contrast detection ganglion cells. And we published a paper in 2014 in Neuron that melanopsin knockout animals have contrast detection deficit. And believe me, this paper gave me more headache. Like, I hated that I published this paper in Neuron. So many people in the retinal field, every time they saw me, they said, we don't believe this paper, this is crap. We know this OKT is only driven by direction-selective ganglion cells. How would you think even that melanopsin is playing a role in this? They ate my head about it. So I was like, published it, didn't talk about it at all, till Kat came in and she said, let's see what happens if we manipulate the M4 directly. Here it is. If you now add the inhibitory dread in the presence of melanopsin, in the presence of normal function, just you inhibit the M4 cells melanopsin signaling, now you get the same deficit in contrast detection that you get in melanopsin, exactly the same. And if you activate these cells, surprisingly, you don't disrupt the pathway, you actually enhance the contrast detection in these mice. So it means that melanopsin is not contributing directly to the brain, but it's modulating the responses of these M4 to the rod and cone input, which then somehow, we still don't know, contribute to contrast detection for the optomotor response. So now we add a new idea to the field that iPRGCs are not only non-image forming, but they could also play a role in image forming functions as well. And with that, I would just like to thank the people who helped me do the stuff in the outside, the funding agencies on the outside, all the collaborators which I thank across the way, and the funding from the taxpayers and all the amazing American people that always allow us to do experiments despite our bad government. And I'll take any questions.
Wow, this was so beautiful. Thank you very much. I have a simple question, how you relate those very few cells in the retina, those RGCs with different pathway and so on, how that relates to human when you have either age-dependent or glaucoma, for example, decrease or attenuation of the number of those cells, because the effect could be quite dramatic since there are so very few of them. Can you, in general, comment on that, please?
Yeah, so this is a wonderful question. So one of the things that we notice for the M1 that project to the SCN, they are highly resistant to degeneration. So my prediction is that in glaucoma, they will be the last to die. But this is hand waving. So most people with glaucoma don't seem to have sleep-wake problems. So and the reason, and this is also hand waving, we've seen it, the M1 cells don't follow the ganglion cells. They actually have axonal connection that goes in the retina as well. So they may get trophic support from the retina and the brain. So they have a dual support. Now, interestingly, I heard from somebody that in a meeting, a group in Australia have found that even midget cells may express melanopsin in the human retina. So this vision stuff may be also important for the human retina. So it may be there is a lot in common, more in common between the mouse and the humans than we thought. So I think, I think humans who work on glaucoma, I would love to collaborate or, just advise without, because I think that's the question we need to ask, Chris. Honestly, it's not been studied very well.
Hey, Samar, that was beautiful. Thank you so much. A lot of really cool work. Two questions. So for the first part of your talk, you showed that if I follow that light induces sleep or body temperature, and that's mediated by melanopsin, because if you knock it out, this goes away. So my question is this a matter of, because the rod and cone inputs will still be there, is this a matter of the light intensity? Or basically, why doesn't it work even though the IPRGC should still be active, right?
It's a fantastic question. So one thing I didn't tell you is that circadian photoentrainment literally can work under the smallest part of the moonlight. So you literally can drive circadian photoentrainment with even weak moonlight. And if you knock out the rods, actually Russell Foster was wrong. There is a sensitivity difference. So it's not true that in rodless, coneless animals, everything is normal. So the rods contribute to circadian photoentrainment. But for the direct effect of light on behaviors, you literally cannot get any responses even when the cones are fully activated. Like you need to activate the cones maybe 10 times the intensity response. So I think what's happening, because cones don't contribute much to non-image because they adapt, the rods are possibly saturated.
Okay.
That's a hand-waving response. I would love for you to figure this out.
That
Yeah.
makes sense.
Yeah.
And my second question is, have you or anyone else looked at the interplay between the melanopsin system and the circadian clock, things like BMAL, for instance? And can you modulate the melanopsin response in mice or animals that lack circadian rhythm?
Okay. Honest to God, one of the things I'm dying to work on is retinal clock, but I'm so scared. I think retinal clock have destroyed so many careers because the retina is so complicated. Retina is a little brain, and the clocks are so many different phases, and I don't think yet we have the tools to figure out how to manipulate the retinal clock independently. I don't have the guts to work on retinal clock. I would love to have the guts, but I don't have it yet. So I don't have an answer, unfortunately. Yeah.
So we have a question from Zoom. Shailesh?
Yes. Hi, Samer. This was a great talk.
Thank you, Shailesh. Yeah.
Always been a fan of your work. So I've followed your work mostly. So, but I'll ask a question with your latest publication in Nature. So this study is in mice. It's nocturnal, obviously. So do we know anything, or can you speculate anything about the human eye cells or IPRGCs? Do they also shut themselves off in a bright daytime, and in the same way, like, or there are special wavelengths like violet light could really shift the human body clock during the daytime? The question I'm asking is because it could have implication in treating jet lags or shift workers. So could you talk a little bit about that?
Yeah, so just if people didn't, so the biggest problem we have in treating jet lag is if you have to treat people jet lag before they travel, you have to really shine light at night when they are sleeping. So you're gonna affect their sleep, and that's counterintuitive. But if you have a way to shift the clock in the day, then you actually could shift yourself during the day before you travel, and that would be much better. So is that found in humans? So Michael Doe's group found the first depolarization block in mice, and then found that it's found in monkeys and human retina. So this is found. In a human, we have a dead zone, is not as defined as the mouse. And the reason is diurnal animals like us, like the people who really are exposed to the more sunlight in the day, we have two forms of entrainment. We have the phase response curve, but we also have what is known as parametric entrainment. So I think we have the depolarization block, but we also have the LGN pathway, which measures photons. So we may be more resistant to like changes in the environment. We possibly will not be as affected as the mouse if we don't have SCN gate, but I still think it's gonna work, and I'm already starting in collaboration with a human group at Johns Hopkins to try to see if we can phase shift the clock with a short wavelength light in a human. Because a human's cones are not UV, we're gonna use something like 415 or 420 to see if we get the same response. So we'll keep you posted, and we'll see if we're able to do that.
We have one more question from Zoom. Eric.
Thank you, Doctor. I'm with the Choroideremia Research Foundation, a rare inherited disease where we lose our vision from out in the periphery to the center. I only have 1 degree of central vision in one eye left. And I'm wondering, is it just the neuroplasticity? I'm sorry, I'm getting tongue-tied here. Is the brain able to adapt to an ever-diminishing signal for this rhythm? Because we're getting to the point of minimal retinal cells, especially no rods and very little cones left. And I'm wondering, is there a minimum level, or have you studied any of that, of how the brain can adapt and just get a little bit of that signal to keep the rhythm?
That's really a hard question to answer. That's a fantastic question. I need to read more about this. I would be very worried if the periphery, because these cells are, I could go back to the retina. These cells are more in the periphery than the center.
Okay.
In fact, in the fovea, they are fovea and parafovea, they are completely absent. They are really avoiding the fovea and parafovea. So that's, I haven't ever heard of like this issue before. So I'm gonna have to say that I don't know. I'm sorry. I feel bad that I disappointed you with the answer.
Oh,
but,
no. Thank
yeah,
you.
sorry about that. Yeah.
Hi, Dr. Hattar. Fantastic work. I'm Sherry from Cognitive Science, and my first question is, perhaps I missed it. Why is it that sunlight does cause the depolarization block, but just simple UV and blue light does not?
So we don't know the exact reason. We think it's all about the strength of the phototransduction current. That somehow, if you stimulate the melanopsin phototransduction pathway very strongly, you get the sodium channel to become highly inactivated. So if you look at the cell, it's highly depolarized, but it doesn't fire action potential. So it's really depolarized, but the brain doesn't know about it. So I think we just got the sweet spot. But I'm wondering, like, why do you have a different circuit? I'm wondering if there is events in nature where we're trying to figure out where you could have a situation in nature where this circuit may be important. And the reason it's involving the LGN, it's also interested that time-restricted feeding use the same one. So I'm wondering if it's under stress condition where the animals have to start going out in the day if they are nocturnal, or in the night if they are diurnal, where this system start breaking out the gate so the animals move against their will. So that's something we're thinking about, but we think we just hit the sweet spot. We were just lucky, honestly.
So where I'm going with this is actually I'm thinking about whether does exo length elongation would be affected.
What, sorry?
Exo length.
Exon length.
Myopia development.
Oh, yeah. Oh my God. Myopia is so interesting because like every opsin that is non-emis seems to contribute to myopia. And it's so interesting. I honestly, I'm scared to talk about this because I know very little. Sorry about that. Again, not satisfying. Sorry. Yeah.
Hello. Hello. Thank you for the talk. So I know this has happened to me quite a few times. I'm sure it's happened to other people where you go home, the light is on in your house, and you sit down on the couch, like I'm gonna rest for a couple minutes, and no matter how hard you try, you're like, I know I have to do something later. You're just so tired that you fall asleep anyways, despite sunlight being outside, light being outside. And you wake up 6 to 7 hours later. That's like a full rest of sleep. So biochemically, what is going on?
Yeah. So because I knew I have so many slides, I didn't present the tripartite, but I would love to use this chance. Sorry, I'm just going to go back to this because I love this tripartite model. I think people have, not that because I created this model, but honestly because I think it's really a way to think about the system in a much more inclusive way. So what I think is happening, I think your point is very well made, and I think that's what I'm trying to say here in this model, is that what you have to think is that behavior is not controlled by one factor. The same thing is when you think of people in the north of the earth in this winter, not everybody causes depression, because you have a circadian clock. So if you have a beautiful circadian clock, then even if you're exposed to the wrong light, you may not be that affected. If you have a beautiful homeostat, if you slept really well the night before, then light may not be affecting you. So when you think of light-mediated behavior, you really you have to think about the circadian phase, the homeostatic phase, and the light phase. So if you're really tired and you have a huge sleep debt, then shining light is only going to affect one of your sleep population. Your other sleep population will override the light, and you will still go to sleep. And the problem, because you have such a homeostatic drive, your clock is not going to be able to keep you awake. And now you're going to sleep really early, wake up really early, and mess up your circadian cycle.
How long would it take to like readjust ourselves?
Sometimes it's like jet lag. Sometimes when students have a sleep debt, they take 3 to 4 days before they get back completely. And that's why the weekend effect, some people call it social jet lag, because people go out and party the whole night, and then Monday and Tuesday they say, we can't concentrate. Surely you can't concentrate. You literally traveled to Europe without your knowledge. You literally traveled to Europe and came back while you're still in your own town.
No more party for you.
You have to live a boring life if you want to be circadianly trained.
Matthew, Deepa, are you ready?
Yes, I am. Great talk, Summer. It was
you.
so wonderful to hear all these stories.
Thank you.
And I have two questions. One is, do you have any insights about if OPN5 is involved in any of these violet light effect?
Oh, that's a great question, actually. I have to say I'm a little bit biased about OPSN5, if I want to be very honest. But now that you mention it, so we're doing the S-cone specific knockout with Wei-Li. If that works, then I would say no. But till I see that, I'm going to say I don't know. Okay. Even though I'm a little bit biased about OPSN5.
That's wonderful. So my second question is, we are seeing that in the dead zone, we don't get any phase shift, but however, do you have any insight about if that is affecting the amplitude of the circadian rhythms? And so is there any broader model for how it's affecting the phase shift and amplitude?
That's a great question. So I did mention, I didn't want to hide that information. I did mention that synapse organization is highly affected by light in the day. And we have been manipulating some of the genes. I'm sorry, I swear to God, I'm not trying to hide, but I could never remember the name of the gene. But we manipulated one synapse gene that is affected during the day, and the whole amplitude and the phase of the circadian system is affected. So it seems that light during the day is not only important, but it's actually part of so it's not important for phase, but maybe important for amplitude and organization of the SCN, which is mind-boggling. So we're working on the synapse organization with light during the day. This is a wonderful question. I'm sorry I don't remember the name of the molecule, but it's CQX something, I don't know. My students always, So you're absolutely right in your thinking,
Thanks so much, Summer. It was great.
Thank you. Thank you. Oh.
Hi, my name is Kim. I'm from Dr. Pacheski lab. It's a beautiful talk. I have one question that would you comment on the senior population where the sleep pattern is dysregulated and how it would correlate it with the PDE10A expression?
Oh, wow. I haven't thought about that, to be honest, with PDE10A. That's very interesting. We assumed always that the older population have a lower amplitude rhythm, and that's why they affect their sleep. But that's really interesting but the PDE10A, I didn't tell you that, but we're actually doing the analysis of the promoter, is active the minute it's expressed, and it's very labile. It's one of the PDEs that is turned out. It's not like the retinal PDE where it has regulatory element. Once it's expressed, it trumps cyclic AMP and cyclic GMP. So it's controlled by the circadian clock, and it has something similar to the PEST domain where it's downregulated fast. So it'll be very interesting if part of the problem, that's a great point. I haven't thought about it. So I can't think about it that fast, but I would think about it more. I love this idea. Thank you. Thank you. I think so. I'll acknowledge her.
Hi, Summer. That was beautiful as always. Question. I think, as circadian biologists, we tend to really think about the clock as the center of everything.
I did.
Yeah. And I think your work really kind of made me think a lot about studies where we've shifted eating, food intake, and this really leads to, obesity, changes in terms of, metabolic rate, all of these things. Given kind of the really provocative and beautiful idea that you're proposing here, right? That we're looking at multiple input cues that are controlling behavior. Your model systems, I think, really set up paradigms where we can dissect control of output behavior and try to disentangle what is more important. So may I ask, in your thought process, where do you put the largest control in terms of behavioral output in food, which I think affects us dramatically, and energy expenditure? Where would you put that control? On the circadian side, the homeostatic side, or both?
So Harris and Maria, who work on sleep, and Maria works on depression and stress, they hate that model because I only put arrows going from light because I'm light-centric, and they want me to put arrows back. But I'm so light-centric, I said, when you go to your labs, you could figure out that question. I still want to figure out the light. You're absolutely right, honestly, because these effects, then these will feed back. They're not just going to be passive to the light input. You're absolutely right. So I'm very interested in the light. There is still tons of questions to answer. But when they get their labs, I am sure they're going to be studying that, to be honest. Yeah.
Hi, Andrew Brown, UCI. Thank you so much. Really inspiring talk.
Thank you.
When people have advanced glaucoma, they have a retinotopic atrophy of the V1 cortex. And when people have a stroke of the V1 cortex, they have a degeneration in the ganglion cells. I don't know if the number of melanopsin cells is sufficient, but if you knock out M1 or M4 or whatever, do you see a degeneration in the brain of the nucleus that they lead to?
We have never looked. That would be very interesting because we know M1 in development, they affect retinal waves. So in the eLife paper we published, if you knock out melanopsin early on, vision is actually affected. So that's why I always mention that we knock them later in development. So it's a really good question. We've never looked. I would love to send you the animals, any animals you want. We always send them freely. So anything you want, just ask me. I'll be happy to send them. That would be something I would love to see what happens.
The reason I ask is because, obviously we can't study these cells individually in a specific patient.
Yeah.
But if you have a surrogate marker from an MRI, then that could be kind of informative about the function of those cell populations.
Yeah, I'd love to talk more. And if you would like to collaborate, not collaborate, just anything you want from me, I'm happy to do. I mean, I would love to know the answer myself, to be honest. Yeah. Yeah.
Oh, thank you. That was beautiful talk. Just naive question. You mentioned something about difference in the humans, especially with the pupillary reflex. And I think, if I'm right, there is in humans versus mice, there is more control in the iris about the melanopsin, right? How does that matter in your context?
Especially that King was my advisor. I have to say this iris pupil, I'm not so sure. Like we have tons of data we tried to repeat. I'm not so sure. It seems in mammal, the iris is not playing a major role. I mean, we've tried, and then I should publish this data, but I hate to publish like antagonistic data, but we knocked out M1, M3 muscarinic receptor to see what is the contribution of the pupil iris muscle, and we could never get any pupil, even with sunlight. So if melanopsin is contributing to the pupil, it requires the normal signaling, which makes it, yeah. And then when you read the literature very carefully in mammals, you find that the pupil circuitry is itself not what is the word, like it's biased. So it's always more toward the ipsi than the contra. So it could be brain reason why you get more ipsi.
But you remember Razvan Gelder had the chicken.
The chicken, yes. The chicken for sure. Non-mammalian vertebrate, the iris plays a major role, and it may be even cryptochrome, which is even more interesting. So flavin and But that's why I get very excited about the mouse. We figured out what muscarinic receptors contribute, so wanted to figure out, can the iris drive full pupil constriction? It doesn't drive constriction at all. If you knock out the M1, M3 muscarinic receptors, no pupil constriction at all. I'm sorry for this, but the human iris is very interesting because the human iris, so the mouse iris is like a photometer, but the human iris is really emotional center as well. So you have to be careful because your emotion, so people are using it for all kind of emotional output. It's an incredible system. And the problem, most of these people, like I'm in the MH, now they ask me about light. They have no idea about light information. Like they think all white light are the same. So I had to explain to them what's the difference of spectra, how to measure light the right way. They think lux is the same for every white light, which blows my mind. But they don't think about it all the time. So for the human, it's much more interesting. I'll be happy to talk more about it for you. Sorry, yeah.
Hi, my name is Rafał. I'm Chris's postdoc. I have a question. What is the complaint of patients that have a loss of function of melanopsin? And if this kind of complaint or phenotype can be compensated by the other inputs to the entrainment?
Yeah, so people have talked to me finally that they have patients that seem to have sighted vision and have sleep problem. So I cannot divulge because but it seems that finally patients are coming in where they seem to have melanopsin deficits, where they have normal vision, they don't seem to have any problems, yet they have sleep problems. So my gut feeling is that a lot of people who have depression, maybe even depression-resistant patient, they may have problems with the melanopsin pathway. That's my gut feeling. But again, we're so busy with the mouse system, we haven't really done as much as we should on the human pathways. Yeah, great question.
So unlike humans and mice, zebrafish, they do have retinomotor movements and they do not have pupillary control. So what do you think, how would it go in them if you knock out all of them, like rods, cones, and the melanopsin?
This is so interesting. We were just in the lab meeting talking about why you have pupil constriction in some animals and not other animals. I don't have a lot of insight about that. So my simple answer, the stupidest answer is the more rods you have, the more you need pupil constriction. But I haven't done a huge research about it. And zebrafish can regenerate, they have more cones. So maybe they don't need as much of pupil constriction as us and mice. So it'll be interesting to look at diurnal animals that have like squirrels, see if they have good pupil constriction or not. So that's something that would be very interesting to look at. Yeah, I don't have a lot of insight. I'm sorry. I know it's not satisfying. I'm sorry. Yeah.
Yeah. Thank you very much for your talk. It was really wonderful.
Thank you.
So I was wondering, do you notice any fitness difference in the mouse models that you generated without certain functions? Or a more broad question is, why do you think animals have like two sets of regulation? What are some advantages of having two instead of just one?
Wow. I mean, clearly I can't do these experiments. I would love to do these experiments. It's something very nice to think about. Yeah, I wonder why you have these different intensity fitness input to the behavior. That's a really exciting question. My simple, very stupid question, because I haven't thought as hard as it is that for animal it's very hard to measure seasonality, and that's why I put the bird example. And it's mostly for seasonality because they don't have calendars like us. But that's a great question to think harder about, and I haven't, to be honest. So again, I'm sorry for being non-satisfying. Yeah, sorry.
So we had a wonderful presentation, and I know
Wonderful
Dr. Hattar,
question.
and heard him so many times. I knew that we're going to be here for an incredible presentation today.
Thank you.
Please join me and to thank our speaker.
Thank you, everyone, for the questions. Yeah. Wow.